Okay, let's start again. Please, Macías.
Okay. I hope you are eating, everyone. So, so I have a lot. Okay, so I just try to keep open your eyes to the end of the story, at least today. Okay, we have more stories every time, and if I have opportunity to show you something else, I will try. Next one would be in Belo Horizonte, but then later on we can be here in Europe, everywhere. So you will see. Well, today, this afternoon, I want to show you something about the search for new models based on structure-activity relationship studies. Okay. I just try to show you some of the examples. For instance, in this case, you have a molecule, okay, and you try to look for molecular interaction potentials, reactivity, stereochemistry, and you try to look for performance and also morphology, charge, performance, and then stability, and based on the chemical structure that you are established. Okay, the objectives, preparation of wheat allelochemicals and their degradation product. We start with bioactivity evaluation or statistical comparison, structural requirement for activity, and we'll see what happened. Could you please? It doesn't work. No. Okay. Selection of the most active structures, modification in order to increase the activity, introduction of functional groups, modulation, encapsulation. So you have many objectives here. As you can imagine, this is not a consequence of 1 year project. It's not the work of one single PhD project. It's a combination of many years and many works together. And I just tried to combine the puzzle to show you something of the story, part of the story. Okay, as soon as you have the encapsulation, finally you go again to the starting point again and try to check again everything. So if everything is working well, you will have on your hands, you will have a very potent tools. Let's talk about Cynara cardunculus. Remember this morning we are talking about Cynara scolymus, and now Cynara cardunculus. It's an example, from my point of view, it's an example in the search for phytochemicals from agricultural byproducts, because this is already used, but know the leaves, know the rest of the plant material, okay? Obviously, we are looking for emerging populations worldwide, and we are trying to go through the zero hunter. We try to avoid synthetic herbicide, at least the traditional ones, because the negative impact that they have in the environment, in the human health, herbicide resistant development. And we are looking for some sustainable development goals, like a good health and well-being, responsible consumption and production, and life on land. So based on those objectives in mind, we are using new alternatives, and it's the case of allelochemicals. In this case, the allelochemicals, we are trying to look for residues from one of the cardunculus. In this case, we have Onopordum acanthum, Scolymus hispanicus, Cynara cardunculus, and Cardunculus variedad silvestris and altidis. Okay, at the beginning is the same story. So you have plant selection to study, bioassay, extract, bioassay, fractionation, bioassay, pure compound, bioassay, and then just try to explain me if it's working or not. We have, as I mentioned, the wheat coleoptile. We cut this part here and here, 4 millimeters, and we take control and the seed germination with the photomed, and we try to see how many seeds are germinated, what is the elongation in shoot and root length. And those are the graphics. As you can imagine, you have here DCM, acetone, ethyl acetate, methanol, water, and this is the positive control. And you have the bioassay with coleoptile, and then some of Onopordum acanthum, Scolymus hispanicus, cultivated cardun, and wild cardun. So you have an idea about what is this. with the experience that you have already, because we are attending the whole morning, could you please tell me which one you will take it for start the work? What extra? Which is the best for you? At least one. Well, you have many, but this is very consistent, okay? Because with dilution, still remain active, particularly this one, the wild cardoon. So, based on this, you say, okay, particularly wild cardoon with ethyl acetate. So you have one fraction that you make the decision to isolate. In this case, you are using Urocloa decumbens and also Echinocloa cruzgali, and try to see how it looks in these two crops. In these two, against these two weeds. This is the control, and this is the extra. You see, the more affected parameter is very clear, is the root extra, the root elongation. Okay, so let's go with Cynara carbunculus. It's a Ceraceae family, wild found in Mediterranean basin, and they have an allelopathic richness, as anti-cancer activity also, anti-VH, antioxidant, cholesterol, hepatoprotective, antifungal, so almost everything can be so wonderful. It's a very nice plant. Do you find something in the market? A lot of products made with Cynara. So you have alcachofi here, you can eat it, very good, that's a very good one. And then you have pills, you have, remember this morning when I talk about pills? You can have pills of extracts, but you don't know exactly what kind of extract is there. You have also Cynara forte, means that you have enriched fraction, okay? But we don't know if I enrich or not. It depends on what is the comparison. Compare me, please compare me. And this is a very typical drink in Italy, Cynar, very popular. It's common here also, Cynar, yes. So the three major components against sesquiterpene, okay? So you have grosschemine, agerin B, and cynaropicrine here. And listen, 94.5 milligrams per gram of dry weight. This is really important. So the concentration of this mixture is really, really very good. They have also polyphenols, triterpenes, and so on, but we are concentrated here because this is a major, the more abundant. So let's go and try to see what happens. With coleoptile bioassay, you see here 1, 2, 3, 4 compounds that looks very nice, very interesting. So it's number 1, number 3, number 5, and number 6. So you have many candidates, good candidates that could be used, particularly number 1, 5, and 6, because the amount 1, 5, and 6. Okay, against Urocloa and against Echinocloa. This is something also I want to point it out. When you are running the bioassay with ethyl coleoptile, you really don't know exactly against what is important. I mean, you don't know if it's herbicide, it could be insecticide, antimicrobial, I don't know, okay? The only thing you know is that it's something that should be studied, that's it. But you don't know, at this point, you don't know against what, okay? So you shoot this, you can see very perfectly that 1, 3, 5, and 6, 1, 5, and 6, 1, 5, and 6 are the best one, of course. One time, twice, and so on. And you face again the same problem. You say, okay, sesquiterpene-lactone, cynaropicrine, I don't know if you are familiar with this kind of molecule. When you make a solution of cynaropicrine and you try to solve in water, it doesn't work. You select chloroform, it's okay. Methanol is okay. For NMR, no problem. But for water and for bioassay, doesn't work. So this is something common to many natural products. And you have two options, as we mentioned this morning. You have the possibility to establish solubility versus lipophilicity, changing the molecule and trying to connect with different side chain and try to work with this, or to cross the membrane, just making salt or make any kind of base condition or ACE condition to change it. Okay. In other words, a structural modification. But if you're looking at in nature, what's happening in nature, this is one example that I got directly in my hands a long time ago. This was in 93 published. It was in almost 91 when we start with this problem. It is something that we found. It was a very nice plant growing in Florida beach, beautiful beach. Okay, and the problem was that the major component were monoterpenes. And you say, okay, monoterpenes in allelopathy means should be soluble in water. And the question comes with the publication. Just how insoluble are monoterpenes? The question is, it should be soluble enough, it depending on the activity. If you are active at 2000 ppm and the solubility is 3000 ppm, it's not so big solubility, but it's enough. Okay? So we demonstrated in this paper, we received the best paper of the year in ecology, because they change our concept that monoterpenes usually are not involved in allelopathy work, because they are volatile. Okay? So at the same time, I remember I was working in the bench. I leave the solution with the monoterpenes in the solvent and so on was not so good, and by mistake I have a solution of ursolic acid also present in the plant in large amount, particularly the leaves are plenty of ursolic acid, and I had my solution there, and by mistake I put the solution of monoterpenes into the ursolic acid. Was completely clear, transparent, no problem, was completely solved. We say, what happened? Oh, a mistake. Let's try, but I say, remember, science, if you are a scientist, you are curious and you observe, okay? And you say, hey, wait, what happened here? Let's try it the other way. I will put that solution of ursolic acid, I try to put it, it's solved. Okay, what happened at the end, at the other way? If I just add it, drop ursolic acid, it's become clear and the solution was plenty. So we try to analyze by UV also, and we know that we established that they prepare micromicelles, as you can see here. And the micromicelles face the monoterpenes in one direction and the polar in the other direction. So what means this? This means that you have the solution soluble in water for a longer time, and you make possible that the activity are related with the monoterpenes rather than any other compounds in this particular case. Okay? This was the origin of one of the techniques that maybe you heard some time ago, is electrophoresis, capillary electrophoresis. The electrophoresis capillary, usually what they do is working with molecules that have positive and polar and not polar sides, okay? But you have molecule like this, monoterpene. Can you apply electrophoresis capillary? In principle, no, because they are not polar. So what they do is, using our technique, prepare a micromicelle and then running in the electrophoresis capillary. Okay? This was the origin of the technique. So my philosophy in life is this one: learning from nature. So what we do is, let's say, there is an example of polysaccharide or any kind of polymer that can help also for the solution or not? And you say, yes, they are. They have chitosan, is a polymer, polysaccharide, azemanan, belonging to aloe vera, okay? Hyaluronic acid, of course, hyaluronic acid. You have hyaluronic acid in shampoo, cream, many things you have hyaluronic acid, okay? And in the case of aloe vera, I was working also many times for aloe vera, we are working with aloe emodin, which is a very good anti-cancer, anti-colon cancer. Excellent, one of the best colon products that we have. What happened is that the aloe emodin is making an encapsulation with azemanan It's a very peculiar polysaccharide belonging, as I mentioned, from aloe vera. So in the case of aloe vera, it's inside, it's prepared. And the aloe vera is moving into the body to bring the active ingredient to the part of the body that need it, okay? When it's touched, like a liver, or like it could be even the brain, okay? Cross the membrane, go through the brain, open it, and then act. So it's making possible. In this case, it was for the colon cancer. Perfect, wonderful. So you have a very good idea that why we can produce it or not. It's in our hands, or it's just a fiction. We'll see. We are using at least 4 methyl organic frameworks. Is the one because they received the Nobel Prize this year. The three people, one from Japan, another one from Canada, the third one from the States. In the middle, you have organic nanotubes, supramolecular micelles, and polymeric nanoparticles. Okay. So we are using this because of what? Because we want to know if we can encapsulate, but with control. That's the question, okay? In the case of nanotubes, you have cyclodextrin, as I mentioned before, alpha, beta, gamma. You have also glycolic acid in the case of nanotubes, or pluronic acid for nanoparticles. And you have here, in the case of of the sesquiterpilactone that we are preparing, okay, sesquiterpilactone, we have different ones with different size. You see here, really big, okay? And we are preparing the nanoparticle for this. We check agarine B, cynaropicrin, grosamine, and we try to check in the etiolated. You have with different cynaropicrin DMSO, cynaropicrin with nanotubes, and you can see that cynaropicrin originally with DMSO, okay, but with nanoparticles even better because with lower dilution is more control. Okay. This is really very interesting. The question again is the joint action. In a plant, usually they have these 3 in very large amount. There is any possibility to have a kind of synergistic effect, antagonist effect, or what? And we test it and we say, okay, let's try. So we choose 17 binary mixtures and try to see what is the best combination of both, or the combination of the compounds. We found 7 synergistic interaction, and we found 2 antagonistic interaction. So it's good because you know exactly what you can mix it or what is better than don't touch it. Okay, as usually the British say, if something is working, don't touch it. But if it's not working, that's the way. Okay. Secondly, can you produce enriched fractions? So in that case, you don't need to make a finally isolation. Just increase the fraction in the compounds that you really need it. Okay? So let's try, and we use it. We use the botanical variety, Globe artichoke, cultivate cardoon, wild cardoon. We are using 3. Okay? And we try to see how is the best for this experiment. Is the wild cardoon. Okay. We try the harvesting is in November, January, April, so you have 3 times where you can collect it. This is also really important because you say, okay, it's very rich, everyone, okay, fine, but how many times a year? Only 1. Oh, you can imagine to move the whole production just for one thing that can be the production in 1 year. If the year is not so good, what happened? Lock, cross the company, okay? Okay, so you have the enriched fraction. You have, of course, the control. You have we have the methodology, okay, and we are just maybe one of you, many of you can identify because you can see the direct connection and injection of the fractions, and you will see with the triple quadrupole we just try to see how is the concentration of the enriched fractions. Okay, we just make a calculation of everyone, so the regression coefficient is very good. And we know for sure that we can make a continuous control of every sample in the mixture. Okay, so what kind of extraction? We propose ethanolic pulsed ultrasound assisted extraction method and use the membrane process technology. With this membrane process technology, what we do is the ultrafiltration. It's another one. Is try to avoid any kind of chlorophyll. So we remove the chlorophyll from the system, and then we have a very clean compounds. Usually, when you are trying to remove chlorophyll, what do you do? Chalcone. You mix it with chalcone, okay, and then cook it and remove it. But you lose also a lot of compounds. So it's much better to use this. So you have this option, membrane process, or the ultrafiltration membrane. This ultrafiltration membrane is the one that we are using, and we published recently, last year. We're using particularly 2000 Dalton for the control. Okay, so you have a very nice thing. Do you know what kind of molecule you want? Do you know the mixture that you want to use? Do you know the fractions you want to have enriched? And then you have also the way to control the quantity of the compounds. So phytotoxic activity evaluated against 9 wheat species, a panel of 9 ones, Trifolium repens, Polaris arundicaria, Portulaca oleracea, and you can see, for instance, in Portulaca, the inhibition of the roots and also the shoot was excellent, really excellent. Then we have also the bioassay in hydroponic control, and it's working very well. You can see the inhibition. All the fraction, enriched fraction, okay? And, well, I'm sorry because the picture is not so nice, but means degradation, nothing else, okay? Nothing else. So we try to choose also the possibility to say what happened if you dissolve it in water and then put it in the media. Maybe degradation, suffer degradation, we don't know. So let's check. So we study the degradation and what is the transformation they enforce. So to evaluate the degradability of cynaropicrine, mainly sesquiterpilactone leaf extract, we choose the cynaropicrine, water pH 5 and 8, commercial soil, incubate at room temperature, and then a cell quantification. Analysis were performed using USPLC MSMS triple quadrupole. Okay, degradation studies show that the temperature is okay, it's a factor, but the pH is even worse, pH 5, pH 8. So you must take in consideration the pH, because in the case of cynaropicrine it's okay, but the sacil cynaropicrine is not so good, depending on the time, you know. This is 0.5 hours, 1 hour, 24, 48, stable with the time. In the soil, you have almost the same, very stable cynaropicrine, as you can see in the picture. And also in the coleoptile bioassay and Portulaca, you have again and again cynaropicrine, desacil, dihydro, and 4,15 dihydro. 4,15 dihydro cynaropicrine, this diethyl cynaropicrine is one of the degradation products. And this is relatively obvious because you can suffer saponification here of the side chain. But what means is that even the degradation product is good. I mean, can be used. It's not something that interfere in the bioactivity. So Cynara cardunculus as a source of allelochemicals is okay, based on the agarine B, grosschemine, and of course cynaropicrine. Cynaropicrine again, alpha, beta, and saturate, alpha, beta, and saturate. 2. Agarine, alpha, beta, and saturate, alpha, beta, and saturate. 2. Michael acceptors, Michael addition acceptors. Phytotoxic and synergistic effect. As I mentioned, we have 6 mixtures where the synergistic effect is better. So don't worry. 1 + 1 is more than 2. This is really very nice. And of course, the optimization of extraction formulation. I think the membrane process, firstly the extraction with the ultrasound, and then membrane process technology to purify. And of course, finally, the encapsulation of the sesquiterpilactone with lithocolic acid nanotubes. In this case, with the degradation studies, as I mentioned, with the pH is stable at 50 degrees at pH 5. In soil at pH 8 is degrade, but the degradation is equally or more active than the ensured phytotoxin. Obviously, the formulation enriched, then we use it greenhouse, and we use it the greenhouse and we test it, we test it and it's working very well. So now it's in the process to be in the field. The greenhouse is okay, but the field is the real proof, okay? Okay, so let's move with benzoxathinoid in the system. Remember, BOA, DIMBOA, and so. And say, oh, Francisco, again, BOA, DIMBOA. We'll see. I try to show you something else. With maize, you have DIBOA, rye, you have DIBOA. DIMBOA. And the major components, Okay. You have one family selection of the starting material, you have a collection of benzoxaphenones, then you have benzoxazolinones, then you have afenophenol and aminophenoxathins. And obviously, you have the possibility to have synthetic analogs. Okay, so based on the results, we decide to use D-diboa. D-diboa, why? Because it's a very simple molecule, and we can modify. We have many options. Organic chemist, play with chemistry, okay? So you have the possibility to have here in C2 position, so you have compounds without functionalization, so you can use C2, benzoxathinoids. The OH could be adding new groups, so you have functionalization of the N4. Then you have also aromatic position free, so you can combine. But in addition, you have the hybrid, so you can change here and here, here and here, and here and here. So you have a very nice combination that you can use. This is the first. The first combination, what you do is just try to modify the OH in the 4th position, and then with different side chain here. Based on this, you have modification, you have changed the activity, but also, which is really important, you change the log pH. So mainly log pH means that the solubility is different of the different compounds in water. And I just want to show you one of the graphics that I really like it very much, which is the case, the case of the chlorine compounds when you change the aromatic ring. You see here we have compounds D-methyl, dimethyl, dimethyl-O-CO, difluor, 7,8, 7,8, 6, and so. So you have many options here moving. And you have that one of the best is 44 micromolar, but also have a selectivity of 39, I'm sorry. Let's change the hybrid. The hybrid is mixture this with this. And I want to show you one of the graphics, I really like it very much. Specificity. Okay? So, what does it mean in this case? Don't forget that we want to have a very nice production, so the main crop should be safe. Okay? But you must remove the competitors. So in that case, what we are looking for is something that you say selective index, which has, for instance, in this case is rice, and then echinocloa. Echinocloa is the wheat, okay? So if the rice divided by the echinocloa is better than 1, it's more active on wheat. If it's less than 1, it's more active in crop. What is your meaning? Means that the selectivity index you want to have as big as you can. Okay, that's the question, that's the message. So you say, okay, let me look here. So I found many numbers, but you have here 8 chlorine, 2 propyl, What is this? 13.39. 13.39 selectivity index. It's very good. 8 chlorine, 4 propyl, and this is 15 or 13 something. And my question comes in the following thing. What do you prefer? To have a very selective index or to have a very low concentration for the activity? Well? Select. Any other? Select. It's much better to have less sensibility with the concentration, but to be more selective. So you have here two options, okay? Here in 2, here in 4, you have a IC50 is 70 micromolar. Here IC50 is 56, not too bad, okay? But this you have 13.29, here is 13.08, so it's much better to take the first one. So you have a list of compounds, you have a list of samples, you have a list of results, and you have a conclusion that you can take it. Now we have 16 patents. I'm sorry, in the group I have 18 patents, but from the 18 patents, 12 are belonging to this set of new collections, okay? Benzoxaphene modified providing interesting leads, obviously. The compound affordable, easy obtained in large scale. This is important to have in large scale. And you have the first level of modification, the best one, the IC50, 28 micromolar, 28. Second level, modification in C2, here, the IC50, 39 micromolar. Modification here with the aromatic ring, 8 micromolar, 2.8 micromolar, 44 micromolar, but selection index less than 1. That's not good. Less than 1 means that you can destroy the wheat and also the crop, so you don't like it, you don't need it. The best one in this case is this one, okay? Well, you have a 44 micromolar, but you have 1139 in comparison in this third level. Even more, fourth level of modification. You have 6 fluor, 4 propyl, and then DDBOA. So you have EC56 micromolar, 56 and 114 micromolar. But in this case, even if this is bigger, because it's 14.4, obviously 6 micromolar with 13.5, excellent. So no discussion. You must to make a compromise between both, okay? What is much better? Because if you put more and more amount of compound that you needed to put in the field, more contamination you are providing, okay? So from this selection, level 4, this is the best. Well, I tell you, this is the impact. The case of benzoxaphenoids, we tested. We don't forget that we have a half-life, as you can see here, very short, 28 hours, 31 minutes. And you have APO, APO, IDBO, and so on, with coleoptile. So let's go to check also with barley and wheat. This is one of the most serious and costly wheats annual winter cropping system. And I want to remember, to keep in your mind, that was very nice to have this one. If you had the acetyl derivative here in NH, you destroyed almost the activity of APO. Okay? This is really important. So let's go with this in the field. This was in the field. Okay? You have the field trial of allelopathic cover crop. You have a proceeding a tomato crop in biological farm here. A plot with rye, mulch left on the soil surface, so you have here. On the rye control, the plot cover crop split, so it means that when you have the cover crop here, it's similar than you have a control. It means that it really controls the system. in the field. What is the mode of action? You remember me that sometime this morning, what kind of proteins, what kind of enzyme I must to check, okay? This is the mode of action of these benzoxaphenones. This is the donor plant, the donor cell, okay? This one, donor cell. And this is the target cell. This is in the mitochondria. This is the root exudation, go in the vacuole, I'm sorry, and this make it outside, okay? And then go through the vacuole here, and then try to destroy the system here. So the absorption is directly to the vacuole, and to the nucleus, I'm sorry. This is from the vacuole and go through the nucleus. So you have a very nice interaction in this case. What do you mean? You mean that you must be precise with the doses and to be precise in the solutions. This is the system. To do this interaction and uptake, okay, it should be open a barrier, and the barrier is related with HDA6 in HDA2. So you know, as I mentioned this morning, if you know the protein that you are looking for, or the system that you want to be protected, you look in the database, in the data bank, and check what are the best. And you say, okay, I have this and this. So based on this, what you can do is take it, okay, and try to see what are the mode of action that is a complexation with zinc cofactors in histone deacetylase, HD. So you do the silicon studies, and you be sure about the complex is possible, what part of the complex is made, where is the site. Secondly, for how long is stable the complex. Because based on this, you can imagine what is the real activity and what is the mode of action and what is, let's say, the molecular way that is working. Okay, APO. We know in a simple synthetic process how we produce it. We can produce it in large amount. APO. Kilograms if you want, if you need it. Okay? We have control of the activity. We have a multigram scale, but we have again, what happened here? Low aqueous solution, low aqueous solubility. And this is really very, very difficult because when you try to say, okay, 5 milligrams in 10 liters, in 1 liter, you try to dissolve it, doesn't work. Give you emulsion, but no solution, okay? So it was something that we are working many times, facing the same problem, one time, twice, and two, and three. I want to mention again, Susumu Kitagawa, Richard Robson, Omar Yagi. They are the three that wins the Nobel Prize because of metal-organic frameworks. In that case, we are using zinc as a metal-organic frameworks. And we use lithocholic acid, which is something that we are producing also in our liver, okay? So it's something that can be easily absorbed. Actually, the concentration of zinc is not so high, it's relatively low, okay? So it's not toxic anymore. And in addition, what we have is that, you know, the mops, what they do is the metal is making, it's like a house, making boxes, okay? The metal organize the linkage and organize like a boxes. And the boxes depend of the linkage, of the organic linkage that you are using, and depend on the metal, of course, the coordination. So in that case, we can decide what is the site that we need it for APO or any other, okay? So this should be very precise. This is because they received the Nobel Prize, not because it's possible to do it, no. It's because the application also that they face. The first one, Susumu, was more concentrated on volatile compounds, gas in general, gas. The second one, Richard, it was most concentrated in catalytic compound, okay? And the third one, Omar Yagi, was more concentrated in bioremediation. So you are a toxic agent in your body, okay? I put your mops in your body, and the mops is catching exactly the compound based on the size of the room, okay? So it's important. But in our case was before the Nobel Prize. We do the same in different way. The same philosophy, but a different way. We try to keep inside herbicides and try to open the herbicides. The second one, organic nanotubes, I show you with the sesquiterpilactone, supramolecular micelles, and polymeric nanoparticles. I want to show you something else. And the new one that we are using also are what? Is a cucurbitans, cucurbiturils. Cucurbiturils is like a donut, okay? And in the center of the donut is fixed the compound. Those cucurbiturils, they call it cucurbituryl B7, here APOCB. You see the APO is placed in this way. And you can see, based on the number of molecules that you are using, connecting here, you can be 7, 10, or whatever. And it is something really very interesting in this particular case. I will show you why. Because the condensation units that we are using is urea. Urea is very popular, very, very common, no problem, it's very well absorbed. Okay, cucurbituryls, N in this case is 7, are synthesized by condensation of urea units. They can be consistent with between 5 and 10. There are many possibilities, okay? But in this case, only with 7 is enough. What's happening is that the APO is protonated in this NH, in this N, nitrogen. What is the significance? What is the consequence? The consequence is that if you change the pH, actually you are changing the properties, because you can open the system in AC condition, but no at 2.5 that is normal, but only 4. I'm sorry, I will show you exactly what is the pH. And also we are using not only APO, so also some mimics, mimics, I say yes, disulfur mimics this NaH that prepare this one and this one. So it's very interesting. I will show you now why is this important. It's modified the pK of the molecule, guessed it, the pKa. This property allows interesting application. For instance, in this case, the unusual the solubility is increased after protonation, and this usually happens at pH 2.1. However, when this is encapsulated inside, the pH is 4.2. Means that can be applied on irregular crops and then open it in a regular way, because pH 2.1, not so many things can grow, okay? Only a few bacteria. Well, in other words, you have here aminophenoxathinones, disulfur mimics, and you have the cucurbituryls, the absorption by plant cell in disulfide mimics and hence through hosting with cucurbituryls. So you have at least 5 systems. I will show you polymer commercially available with this generation of nanostructure here. This thalamide, remember this morning, the thalamide? And then using the polymeric, pluronic, you have pluronic, and you have polyvinyl alcohol to avoid the aggregation, and you introduce APO, okay? This APO is very nice because you can follow the structure. You must to convince me that the structure is correct, okay? And I do it by electronic microscope, high resolution electronic microscope. So this stem, they show you here, you can see the holes. And you can see it's like sponges, like the whole of sponges. And every sponges can allow to go in APO. These nanoparticles allow an enhancement of the water solubility without structural modification. So, and we try to see if it's there inside or outside. We have advantage. Why? Because we have nitrogen in the molecule. So we have nitrogen, we follow the nitrogen, and the nitrogen is only inside the particle. Okay, we checked the particle with SEM, and we follow that the nitrogen is only there, inside, not outside. This is the image that recreate the 3D shape of nanoparticle synthesized by combined transmission and scanning microscopy. And you can see how it's like a banana, okay, with the different holes, and the holes contain the nanoparticles. Okay, in other words, the solubility of APO increases more than 25 times. It is unbelievable. Okay, how can the knowledge be applied? The best question. Okay, you tell me many things. Okay, but in real, what happens? Let's see. I don't know. I have agro application in that case because we have a very nice agreement with Sicily, and we choose 3 plots, different plots, and try to see how it looks, the use of these compounds. We shook the biomass, the width species richness at 3 different locations in Sicily, in Italy. We analyzed using the 2-way analysis variance. So you have in one, we apply 1.5 grams per square meter, and the other one was 0.75 grams for square meter. We choose there, and we can see that the overall T2, I mean the second treatment, the best result in terms of productivity was minus 57 with biomass when compared with control. 57 or reduction. You say, well, no, it's a very good number because it's combined with an increase of the production of 45%. So you increase the main crop 45 and decrease in a 57% the biomass with biomass. So T1 and T2 did not cause shift in wheat communities. It's important because the community, the biodiversity is the same. Means that it's okay, it's controlled, okay, but not removed everyone. Moreover, the nanoparticle containing formulation give reproducibility result across the 3 locations. So we are applying and we calculate it and it's working perfectly. Okay, this is something also very nice. When I was talking with my student the first time, we tried to work with nanoparticles. We are not dedicated the whole day for nanoparticles, but we can do it. There are a lot. I was asking for many questions. The first one is to be sure that the nanoparticle was formed in nanoscale. Okay. Secondly, where are the encapsulated compounds, inside or just outside? Third, concentration, to make a calculation for the bioassays. And 4, and it is very important, four, but they are number 5. 4 is how I can remove it, liberate the active principle. And the 5, which is also very nice, is how is increased the solubility in water and the viability in the media. It could be in agriculture, it could be in drug systems. I can tell you, all the system I show you, I have the answer for those. So I know the instructions also. And the question is, when I recommended one, or when I recommend the second one, or the other one? It depends on the molecule, it depends on the ecosystem, it depends on what is the problem. So tell me your problem, and we can prepare exactly the best option in that case, okay? So I tell you, biocommunicators have application as herbicide and drug active ingredients. Full organic formulation process increase the solubility with great application as herbicide. Furthermore, their viability is also enhanced by encapsulation. There are a huge number of nanomaterials and new encapsulation techniques available to be applied for bioactive compounds, not only in agriculture, and we are working on it. In vitro, results are really promising, but now we have demonstrated that field application is also possible. Promising advances have been done in the pharmacology research line with demonstration of activity focused on gastrointestinal system and pancreas and so on. But the silent war continues. We'll see what happened there. And I don't know, but in the future, a Nobel Prize maybe recognize the science behind the secret language of plants. Meanwhile, the silent war continues, obviously. I think we are working many years on this, as you can imagine, as you can see. This is only a brief message, but we are working in many fields, many things, and I tell you, this is something that you will see the application in a short term from now. I'm completely convinced that you will be the receptors of the idea and the development of the system. And don't forget, please, secret language of plants is important. Okay, if you remember naphthoquinones, when I mentioned teca, I have another kind of naphthoquinone. What kind of naphthoquinone? You know this? This is Juglans regia. So you have jugulon, and you have also naphthoquinone that produce apoptosis, control, phytotoxic effect, anti-Leishmanian. Oh, sounds good. But you have also Lawson. Lawson is very similar. You remember? You see? The difference here is on OH, nothing here. Jugulon. Lawson, the OH is not here, it's here. Just a single chain. Lawson, okay? What is this? Lawsonia inermis. What is this? Henna. Very common, very used. And in north of Africa, plenty. They are used many times. But at the same time, they have a very anti-inflammatory effect. This is important also. Okay, so the question here is, let's say preparation of esters and ethers for varying chain length, biological characterization, determine the structural physicochemical requirement, and see what happens. So simple. You have an OH here, side chain. You have an OH here, side chain. At the beginning it was very simple, at the beginning. But then you see, okay, there is one of the collection, very good. So you have 1 more, 3, 4, 5, 6, 7, and so on. But in this case, they are not so good. I mean, you have sometimes problems with the acyl derivatives. In this case, you have Logrand with the weak coleoptile. You have the ether, no ester, but ether here. You have also very good correlation. But in the case of Lawson, you see here, you lose the activity. So we have the correlation, and it is a curve that they show you the activity is a function of the structure, of course, and also the requirement that they have. Okay, log P, and also the number of functional groups that they have. Okay, in this curve you have the best one are located here, and with 5 or 6. 6 maximum, 5 to 6 is okay, it's working well. The side chain. Phytotoxic, you have germination, inhibited, root length, shoot length. Oh, this sounds good. You have increase, 1, 2, 3, so it means that ST, STS, okay? And the cluster analysis. The cluster analysis, they show you how would this compound 1 to 5 and 14. 1 to 5 and 14. The next level of phytotoxicity. And then phytotoxic biology in Echinocloa, real problem. You have also a very good profile of activity in root and shoot. So acyl derivative of jugulon, the first compound has been described as allelopathic compound, okay? Were tested in biology in general activity. Ether derivatives were strongly inhibitory for both series. A correlation between activities observed with lipophilia. Acyl derivatives of jugulon and alkyl derivative of Lawson were compound most active against STS, and alkyl derivatives and Lawson show a better activity profile. So again, you have many options, many tools in your hands, and the message is very clear, okay? The real application of natural product is limited by the water solution. Don't worry, with nanoencapsulation we can fix it. It is necessary to enhance the solubility of natural product without structural modification. Perfectly, because the nano-encapsulation again. A good idea to develop is in the area of encapsulation process, keeping an organic encapsulator with innocuous or positive properties means that when you have a polysaccharide or you have the linkage, could be also useful for the system. And the nanoparticles encapsulation of strigolactone mimics is an example where the water solubility was increased 27 times. Well, this is only the starting point. Maybe you will think about and try to find new ways. Believe me, it's possible. You can do it, okay? Bioassays development in a scale, just few parameters to evaluate, because we can change the additives, we can change the coadjuvants, and we use only water. The interaction microorganism plant should be implemented, of course, because in the future bioassay to understand the replicate should be done. For instance, with fungi, particularly fungi. And of course, don't forget synergism. Synergism is one of the key points. In nature, everything is in a mixture. When you prepare an extract, it's not a single compound, it's a cocktail of active compounds. And you can see that everything is there, the active, the inactive, and the antagonism. So you must to calculate the best way to use it, okay? Studies about phytotoxicity are focused on natural products isolated from plants. Nevertheless, transport media of these metabolites has been overlooked. Why? Because you say, okay, this is very good and the bioassay is working, should be work. No, usually no, it's not working this way. Chemical modification of initial compound released from organism to the media can change the activity. Can change, one compound produced by plant can be changed by light, photochemistry, by chemistry, because the chemistry is involved in the field. Microorganism could be also changing. Even could be blocked because the production of some complex with humic acid and certain others component of the soil. Okay? So you must take in consideration all the system as a whole. The original activity can be reduced or enhanced because of the modification. Sometimes the metabolite product, the APO, was more active than the previous one, but sometimes it's inactive. Aminophenoxacin are an example of increased activity of biodegradation of DIBOA and DIMBOA. And of course, future studies should assess the importance of transportation media and degradation procedure to get better understanding about real chemical communication among plants and other organisms. Final remarks, okay? Allelopathy remains the reference in studies concerning interaction between organisms and the ecosystem. The concentration of natural products is relevant to define the phytotoxicity, but not the only parameter, okay? Allelopathy is a dynamic process that is extremely complex, that involves time-dependent factors and simple effects, and the analysis of this is not possible, which means detailed investigation are always required. Sorption, viability, and soil roles need to be considered to obtain complete overview. So it is not possible to say this is an allelopathic interaction because I saw this, and this, and say, well, what happened with the degradation, what happened with absorption, what happened with many factors. And the discovery of new mode of action, this is the key point. And the great change of allelopathy has undergone in recent years is crucial to decrease the use of traditional herbicides. Please don't forget something that is also really important. When you have something really valuable in your hands, don't go to publish immediately. No, that's not the way. Firstly, try to protect it as a patent. As soon as you have a code of the patent, publish. Otherwise, it's public domain. And there are many companies just listening to you, to me, to everyone, in the literature, just for new ideas. And if it's published, already published, they don't need to pay anything. So you are working for free to those people, okay? That's not the way. Sorry. This is the part of the group, okay, but they are not the only one. We have many from many countries, and a big group also from Brazil. I don't know, you're here. Have you seen your face here? Maybe, no?
No, I don't know.
I don't know. I just try to find. Sonia is here. Pedro is here. Pedro is here. Oh, there are many here. Those are also from
No.
this is from these two are from
Maristela.
no, see Maristela,
Sadal.
yes,
Pedro.
Fabia, no start, you are here, okay, this is the place where I'm living, okay, this is Spain, is the south of Europe. This is Gibraltar, Strait of Gibraltar. Gibraltar is located here in Cádiz. It's the British side of Spain, you know, here in Cádiz. And Cádiz is this one. And of course, this is north of Africa. So you have here Morocco, you have here Ceuta, Melilla, some other places. And this is how it looks my city. Cádiz is this one. It's a very small one. You see, it's a peninsula. This is a new bridge, Constitutional Bridge. And this is the oldest city of Europe, founded by the Phoenicians 11 centuries before Christus. 11 centuries, yesterday, okay? I just want to say thank you very much for your attention, and you are very welcome every time. Thank you.
Okay, thanks a lot, Macías. I'm sure everybody could learn a lot about allelopathy. And the main, I think we could learn how to work in allelopathy, the method to work in allelopathy, and see so. Thanks a lot.
Okay, thank you so much. Thank you.
Any question?
Professor, you said the key points now in allelopathy is to define the mechanism of action.
Mode of action.
Mode of action. And you bring to us in silico tools to see the mode of action. There are assays that we can do manually to explore the mode of action.
They are systematic. We can do this. It is a protocol, okay? And you can have basically, at the beginning it's very simple because it's only, for instance, do you affect the mitochondria? Do you affect their respiration? Very simple. Why? Because you just can measure, you apply the compound and then you measure the oxygen production, okay? Is the oxygen production, the oxygen production is increasing or decreasing? You can say, okay, maybe it's a chlorophyll or maybe it's a respiration, or you can imagine what a consequence, okay? Or for instance, the mode of action is the disruption of the membrane. Simple, because you just can take a measure of the conductivity. Okay, so if you increase the conductivity, it's because you make a disruption of the membrane. But let's say 7 big mode of action that you can check. And in this way, this is the traditional way. It's like when you are, let's say, looking in a big mountain, okay, for a needle. Where if you are lucky and you put the face in the right way, maybe you have a possibility, okay? But if not, you can lose the whole life and you never can catch it, okay? So I think, from my point of view is, okay, you know what happened because the bioassays that I have run, I have some ideas, okay? But I'm not completely sure. So what I can do is the one that I show you with APO. We know by that time that was related with the enzyme controlling the zinc concentration. Say, okay, try to do in silico studies and try to see if it's working or not. The best thing that we can imagine, that we are very lucky and we'll be, let's say, rich, I don't know, but maybe, is you found a new mode of action. Because at the moment, with this big society that we have, only 20 different modes of action are in the market. Only 20. So why many branches? Why many different herbicides? Because they change the proportion. 10% of this, 5% of this. 10% of this, 10% of this. And they change. Why? Because the money is very expensive. Any ingredients that you want to have permission, it could be 1,000 millions or something like that. It's a lot. Because they must pass to control many things. Maximum now. Because they are more let's say, restrict, particularly in Europe, with the regulation of the new formulation. Actually, glyphosate, we want to avoid glyphosate. But the problem was not glyphosate, what the coadjuvant that is going in the glyphosate, in Roundup is the brand, okay? So, what I tell you, they are a protocol, but if you have any idea about that maybe is something related with photosynthesis, or si it's photosynthesis, check in the data bank and try to look for new possibilities in the silico studies. Maybe it's not necessary to invent the wheel every time, but it's something that we need to have real protocol to establish the mode of action. Indeed, what we have right now is the possibility to say it's not this, it's not this, it's not this, it's not this. So what? It's not this, but what is? You know? And this question is not so easy. We must to find a way to look for. It's a good question.
First, thank you for the presentation. For us, it's very important. When you was talking about rice and wheat species, you talked about selectivity. The question that he did to you, for me, is important because I want to do a question to you about the mechanisms, physiological mechanisms that cause that selectivity. I think that it's important to have clarity which mechanisms are
involved
that cause that selectivity.
Oh yeah. And well, it's difficult to establish, as I mentioned. Yes. Also, you must consider, let's say, Domino's reaction. You know what I mean with Domino's reaction, right? I affecting one, but this affecting two, and this affecting three, and this affecting four. So finally, what is the real mode of action? A combination of everyone. Okay? But one is the first that induces the first action. And this is the question. The mode of action is a critical thing. Is a critical thing. So every people that want to put emphasis on the study of the mode of action, they are very welcome. Believe me, it's really important, very important. Actually, when I show you the way that some of the ethyl coleoptile modify the form, because it was straight, now it is like a banana, okay? They can be related with the physiological function, because the product provoke some kind of malformation of the some of the cells. So this is something that they are, let's say, ideas, but you must to combine the puzzle and try to find it. So I invite you, of course.
Thanks.
I have a question.
Yeah.
Do you think that we are close to find, to discover a herbicide with a new mode of action?
Sure, I'm convinced that. Yeah. Listen, the best and the biggest lab that we have is nature. Thousands of years, millions of years working, and we are the result of the best combination. We are the result. So in nature you have the result of the best combination between different plants, different systems. Why an invader become invader? Because it was not the original ecosystem. If it's any ecosystem, usually survive because the equilibrium. Okay? If you have equilibrium in an ecosystem, in a society or whatever, okay, the society grow up and the combination grow up. But when you have a disruption and some plant comes and make an invasion, is because you have no good relationship with me. Oh, I don't know. What happened? And you change the rules. And this happens usually in nature. So we must take care of the equilibrium. This is the key word, equilibrium. In the world, in our life, and of course in plant cells, in plant systems. The equilibrium. So it is possible to find new mode of action, for sure. Yes, completely sure. Yes. But based on natural things.
Yes.
Learning from nature. Right?
Thank you.
Okay.
More question? I have one.
Okay, let's go.
I'm very curious about the biosynthesis of the benzoxazinoides. It came from antranic acid. I don't know.
Antranic acid, yes.
Yeah.
They are, this is the origin, and they are modified because, you know, it is very well established, the transformation, until found the final benzoxazinoids. And I tell you something else. At the beginning, when, let's say, we start in, let's say, 20 years ago, okay? All the people that were working on natural product chemistry, and you ask and you say, okay, what do you think, when is going to be produced this compound in the plant? Is it in the seed already? Or could be at the beginning of the process of germination? Or is a consequence of the evolution when they grow up and appear the root and the shoot? Usually the people will say, no. Root and shoot, when they appear, is a consequence because the metabolism and so. Firstly, when you have a seed, all the energy, all the nutrients start just to make the germination and to grow up the roots and the shoot, okay? But in this case, these compounds appear in the early beginning. We measure when they start the germination and they are there. Yeah. And you say, why? Mainly for one reason, because they are excellent also against microorganisms. Mm-hmm. So they avoid any infection from fungi, from bacteria. So they give options to the seed to grow up. Okay? So what's the dimension? Yeah? It's very interesting.
Thank you.
Oh, thank you so much.
There is a question. No?
I have one more.
Oh, yeah.
So now, you mentioned in the morning something about AI really quickly.
Oh, yeah.
So Dr. Choi from Leiden University came here a couple weeks ago, and to me it was really clear how he's concerned about how it's changing the research right now since the AI came. So in your work, in your team, how do you have struggling with that or working on it?
Oh, it's very easy, I tell you. I'm an organic chemist, don't forget, okay? So, for me, how I use analytical chemistry, how I use it? As a tool.
I'm sorry.
Tool, as a tool.
Exactly. So
No. Analytical chemistry, as a tool, okay? Because you say, okay, I'm the organic chemist, and I say, I want to use this to combine with this in these conditions to produce this. Okay. Are you sure that you have at the end whatever you want to say that you will have? You need analytical systems to do it, okay? So NMR, HPLC, and so. For me, to develop new techniques for NMR is tools. They are tools. But they are not the idea, okay? Those are my ideas, okay? That's in my mind. And what I just asking you many times, you or whatever, is how I can use it, how I can measure. You can imagine my student when I asked the question and say, please tell me how many molecules do you have inside each nano particle that you prepare? You can imagine. Say, That's a good question, because if I have no idea how I can prepare the formulation, prescription, take 5 grams of the nanotubes and say 5 grams of nanotube of what, right? So the first thing was try to analyze in a very accurate way what is the amount of molecules that you are encapsulated, each particle. You have a per median, you have an average, and you say, okay, the average you have is this one. 30% is encapsulated. Oh, fine. So you wait and you say 30% of this is my molecule. The other one is the the polymeric. Okay, so tool, encapsulation. What is encapsulation? It's a tool. Okay. Why encapsulation? Because I need it. Because I need to modify the solubility. Right. And if I want to keep the compound untouched, I must to prevent everything untouched. It should be encapsulated. Learning from nature, as I mentioned. Learning from nature, I saw that aloe vera produce aloemodine encapsulated. Why I cannot? So I try. So I try in different ways. I try based on the volume, based on the structure, based on the polarity, based on many factors, but I try. And I use it, at least I show you 5 ways that we are using, actually 6 ways. Okay? So, tools. The question is AI. How I can use AI? Easy. I have my dereplication system, okay? Where I compare this and this and this and this and this data with the new ones, this and this and this and this and this. So rather than to make mistake with my eyes that they are very tired, very bad eyes, what I do is I say, okay, please compare with this table. I already prepared the table. This table is not anywhere. It's in my lab. It's in my information. Okay, I prepare this table and say, please, took this information I give you. This is the list, and compare with this. So it's a tool for me. A tool. Okay? Whatever, never at the moment, never I say is, please, could you tell me what is the next project I want to read it? I want to do it. Right? So you must let's say, take advantage of the technology.
I just mentioned because as well, currently, the reports always give some, like Dr. Choi mentioned, your results also plus AI results inside the new reports nowadays is getting more common. This is the point, my question.
Yeah. I mean, for me, it's something that you don't need it. I mean,
Don't need it.
you don't need it. Actually, no.
Okay, sounds good.
It's just, let's say, I want to know, I need the report that you are working, I mean the publication that you send me is original and is not produced by AI, okay? And send me a report that show me that this is not true. Because the English, I pass through AI the correction of the English, of course. I'm not native. Even in Spanish, sometimes I say, please, make a correction. And they give me they polish the text, okay? So as a tool, I mentioned a tool. Whenever I say, please, draw me a report about this. No. Okay? Take advantage. What is the advantage? Remember, if I have to put the seeds, root, shoot, and try to measure with a rule, okay, 4.5, 4.8, could you please, no, this is for, if I avoid this, and I take something that could be simple, very simple, with electronic machine, just make it the point, and make an algorithm that can calculate it automatically, the AI is the same. So use it to save time. Use it to be alive. I mean, to enjoy it with the friends, with the family, and so on. And to be a scientist, not a technician. Technician is technician, and it's needed. But you're a scientist. So what I expected from your side is a critical analysis of the message. The message I show you, but the message that you have, read it in the paper, or you read it in the review, or whatever. If you have clear ideas in your mind, you can fly like a butterfly, for sure. Okay? And please never accept, I mentioned never accept, impossible. Never. When I just start with allelopathy, the people ask me, are you crazy? Are you crazy? Many times. Believe me, but I'm here. And I'm very happy to be crazy.
Lovely.
Yes, please.
Only, one second. exactly he said. In my opinion, you need to know our area. Our organics, I need to know very well organics, very well. They don't understand everything. There are 62, exactly he said. The same in different areas. Agronomy, you need to know about your area. Ok.
Foi a Alessandra Ambrosini, ela fez em português. A pergunta é: dentre todos os seus trabalhos, qual você acha que foi de maior sucesso?
Could you?
Good question. A good question.
That all
All my work, what is the most successful work? Let's say, if I'm looking for numbers, okay, I will tell you was a paper talking about the search of natural product chemistry, of natural products as a drugs. Okay, was published in Research in Nature and Research Drug Discovery. Now is the third year, and we have 4,000 reference citation. So this is in the number point of view, okay? But since the allelopathic point of view, okay, I have 2 pesticide science, pest management science, that are related with allelopathy as a new tool for weed control. The third one was standard target species bioassays. So yeah, we are very lucky with this. And the 4, I tell you, the 4 was something really very appreciated. I don't tell you what place it is, but was here in Brazil. I was visiting the university, and some people introduced me all the stuff, okay? And one of the people say, "Oh, professor, I meet you already." But you know, the name, maybe I forget the name, but the faces, usually I remember the face. Say, "Yes? Where?" It was in Amsterdam, when you received the prize from Rompulin Award, and you changed my life. I say, "Oh, tell me." Because I changed my research point of view. I opened my eyes in a different way. And now I change my research work here in Brazil since that time. You know, that's the most beautiful thing I never heard. Thank you. If I can open the eyes in a different way, that's my pleasure.
And thank you all the people who are attending the course. Thanks a lot.
Okay, thank you.
Okay, now let us talk with your